Okay, we can start. So good morning. Welcome friends back to our regular Saturday Club morning meeting. And today we have a very different topic to learn about and a very different speaker, I would say, because he's a techno-medical person, I would say. So happy to welcome Dr. Siddharth Srivastava. He has done his MSc and PhD biochemistry from our own university, MSc Baroda. He is presently faculty of Medical Technologies program jointly run by IIT Jodhpur and AIIMS Jodhpur. He has a very long CV, but just to summarize that he had his training in University of North Carolina in research in areas of infectious diseases. He also worked with McAllister Heart Institute doing research on uses of interleukin-11 for limb ischemia and was involved in startups which were concerned with education and molecular oncology. So with this brief introduction, I welcome Dr. Siddharth Srivastava and request him to speak about introduction to precision oncology with an interesting CA lung case. So welcome Dr. Siddharth, you can start your talk. We'll have about 5-7 minutes of discussion at the end of your talk.
Thank you so much, Pandya sir, for calling me. And I would also like to thank Radhika Vaishnav to connect me. She and I, both of us, kind of like graduated around similar times from MS University. And Baroda always, has a very special place in my heart. I spent about 7.5 years in Baroda, mostly at MS University in and around. And it was always a very special time of my life. So Baroda always, touches the chord in my heart, and I always find reasons to come back to the city. Today, it's a pleasure actually to talk about this topic. So it's a long journey how I ended up at medical technologies program at IIT Jodhpur as a faculty at IIT Jodhpur. So I'll start sharing my screen and let's go about it. So I'll start with a very futuristic scenario, which actually is not very futuristic. So imagine there is a 42-year-old male patient, right, goes to your clinic and complains about just first and second point. It's like constipation and unexplained weight loss, right? So there could be, and let's say he didn't talk about blood in stool, right, which is kind of like telltale for colon cancers and possibly, right? So let's say you talked about constipation and unexplained weight loss. So if this patient goes to a general practitioner, there could be a lot of reasons why he had constipation, and he may end up just eating isabgol for a very long period of time, before which he realizes that it's not working, right? But imagine a time where our honorable Prime Minister says that Mitro, all the genome sequences of every citizen of India will get their genome sequenced, and it will be like housed in this server, giant server somewhere in India. And any doctor with his or her medical registration number and patient's Aadhaar card could unlock the genome sequence of this patient, right? So let's say it was there, let's say this 10 years fast forward and we are already at that time. And the genome said that DNA sequence, MLH1 PM2060 pathogenic cosmic inside databases. So what does that mean? It means that MLH1 gene, right, it has a mutation or a change. So DNA is nothing but a string of ATCG, 4 letters which repeat again and again, right? So, there is a change at the 26th position where a amino acid methionine converts to threonine, and this change is pathogenic, right? It is pathogenic confirmed by two databases, cosmic and inside database, right? So now we are talking about medicine which has where the genomics has really integrated, and it is being taught at the MBBS level, and the general practitioner also knows about it. So the moment he sees or he or she sees MLH1, this entire line, so the moment the doctor sees that, he knows that, constipation and unexplained weight loss is not because of other reasons, but possibly this patient has colon cancer, and the tumor is somewhere lodged in the rectum or colon, and the family also will have, multiple members of the family will have colon cancers, because this colon cancer is happening because of Lynch syndrome, which is an inherited form of colon cancer, right? So then quickly this doctor will suggest a medical oncologist or an oncologist or a radiologist, like, go there, and based on the NCCN guideline, National Comprehensive Cancer Network guidelines, the treatment starts, right? And if you see the guidelines, and this is an older version, but clearly, like, there is a positive mutation found in MLH1 gene, so see Lynch syndrome management, right? Genetic testing of at-risk family members. So you can see that, how quickly the patient management shifts from something, as simple as isabgol, drink more water and things like that, eat more roughage, to suddenly Lynch syndrome. And and what does it say is that, then the management will say that colonoscopy should be done at the age of 20 to 25 years if the patient was younger, right? Or in this case the gentleman was 42 years, so immediately it will start, right? So there are data to suggest that aspirin may decrease the risk of colon cancers in Lynch syndrome, but optimal dose. And so the point I was trying to make was, the moment you get genomics involved in patient care, a lot of things becomes easier and intuitive, and things may move fast, right? So is it a pipe dream or really happening? So, well, I mean, if you see, six of the countries, and this slide I prepared almost like 3 years back. So Japan, UK, Iceland, Netherlands, Finland, Sweden. So they are already in that process where every citizen of this country is getting their genome sequenced, and that sequence is stored somewhere in the country, and that analysis of that will be available to the treating doctors, right? Now, this is a very often quoted graph, if you talk about genomics in medicine. So, i- this is the cost per genome sequence, right? Which is in green, right? And it is compared with Moore's law. So Moore's law basically explains how computing revolution came. So every 2 years, the size of the chip reduces to half, whereas the processing power of that chip, it doubles, right, every 2 years. That is very evident when every 2 years when you go buy your new phone, that is what you see, that you get cheaper phone, but the processing is much higher, right? So that is a Moore's law. And the cost of genome sequencing actually beats Moore's law hands down. So what this tells you is that lot of genome sequencing is getting cheaper and cheaper by the day. That is why its adoption in clinic is going to just increase over the period of time. And a lot of businesses which work in the area of genomics are going to flourish, right? So this is why the scenario is very hopeful when we talk about precision medicine or precision oncology, that or genomic medicine, that this is going to be happening and it's not going to take too much time. Actually, we are also practicing precision oncology in clinic, and I'll give you an example of that. The third generation sequencer. So currently we are working with second generation sequencer, which are like pretty big. They cost a lot, anywhere close to 2 crores upwards. But the third generation genome sequencers are very handy. This device is only 1.5 lakhs. It's not as accurate, right? And it's not high throughput, but you can keep it in your pocket. You can connect it in your laptop, and you can work. And so you can imagine that these devices are going to become much more accurate and more popular over next 3 to 4 years, and that will democratize genome sequencing. And then you can have, like, PCOs came, and after that PCOs, everybody has a phone, so everybody got the computing power. So similarly, you might have, like, 4 or 5 people who would do genome sequencing services right there in Baroda, right? There is not one person right now. So after these kind of things, there'll be like 4 or 5 of them. So that is nothing but democratization of genome sequencing. So I'm just giving you an idea of how things are progressing. And then, I'll come to the precision oncology so that we can discuss that CA lung case. So precision oncology became a reality because of hard work of a lot of people, right? So every medical or every thing which comes to the clinic, initially it starts in research lab somewhere. So, research labs are working on genome sequences and connecting the mutations or changes in the genome with cancer. So, a lot of these labs in abroad in India were working on that. And so, this connect was established that when there is a mutation in the genome, it leads to cancerous cell growth, right? And then human genome project happened, in 2000 somewhere, 2003 or somewhere around that. 2001, I think. So it was a big splash that, okay, you know what, we have sequenced the entire human genome, very big news, right? But what people did not realize that after the human genome sequencing, there was a much, much larger project of cancer genome sequencing, right? It was spearheaded by the Cancer Genome Atlas project in US and ICGC, the International Cancer Genome Consortium. So TCGA took a mammoth task of sequencing genomes of various cancers and, segregating cancers. So let's say if you talk about breast cancer, then, ER positive breast cancers, HER2 positive breast cancers, or triple negative breast cancers. If you talk about, ovarian cancers or serious ovarian, high grade serous ovarian cancers, or endometrial cancers, lung cancers like adenocarcinoma, squamous cell carcinoma. So segregating and then sequencing to figure out what are the landscapes of genomes in these cancers, right? So once that hard work was done, then a lot of diagnostic labs and therapeutics they spawned. So diagnostic labs said that, what, we can sequence the genome for you, for your patient. We can tell you where the problem lies. And the drug company said, okay, we build the therapies which will attack those cells which with those kind of mutations, those cancerous cells, and you can treat the patient. So that's how everything came together to make it possible that the precision oncology could be practiced in clinic, right? So how did it change the clinical practice, the flow of clinical practice? So one is, when the patient used to come, they used to interact with surgical oncologist, radiation oncologist, medical oncologist, histopathologist, and radiologist, right? But now a new person came into picture, a molecular pathologist, right? So interestingly, we in India we do not have a really good course where a person can learn molecular pathology, right? Because this person has to be expert in both the genomic laboratory science, which is a very it's really an art, so it's a skill. Just like surgeon takes years to perfect the skill of surgery, molecular biology takes years to perfect the skill of doing the techniques, because it is not as automated as clinical biochemistry right now, what we can talk about, right? So and then also this person needs to understand the disease so that the accurate kind of reports could be written, right? So now what happens is, once the patient is with medical oncologist, so medical oncologist wants to understand what more could be done. Can I give some sort of targeted therapy to this patient? Can I practice precision oncology with this patient? So you need to send the report, clinical history of this patient and the histopathology report, to molecular pathologist. Then molecular pathologist then goes back to the clinical oncologist and says that, okay, we did this kind of test, and what we feel is that, for prognosis, the cancer may look like this aggressive, blah blah, or this kind of treatment is possible, right? So then the clinical oncologist can take a decision that, okay, should I go with a certain type of therapeutics or not, right? So let's take, if you have any question, you can stop me, right? And we can discuss, no problems. So let's say, we take a case of a lung cancer patient, and this is the, this guy got into your clinic, and there's clearly, by radiology, you can see that there is a tumor there. So it's an advanced stage 4 tumor, and most likely will follow the NCCN guidelines of 2022. Um, So this was the radiology report and CT scan. So it said that it's a right parahilar malignant lesion. So in the parahilum, and lymphadenopathy. So it has already moved into the lymph system, and lymphangitic carcinomatosis. So really spread a lot. And if you, it's like, bad news after bad news. So mild right pleural effusion with metastasis, right adrenal metastasis. So it has already metastasized. It has gone to stage 4, right? So not a great case to deal with. If you start giving chemotherapy to such kind of patient, the normal overall survival is about 7 months, right? Actually, I took this from one of the classes that I teach in IIT, precision medicine class. So a lot of the times engineering students are not aware of like pleural effusions and all those things, and what is bronchoscopy guided biopsy. So these are just pictures that you won't need. And this was a histopathology report, and it says that it is a, so here it is classified, so it's a non-small cell carcinoma of lung, right? So if you take lung cancers, you can broadly segregate into non-small cell carcinoma of lung or small cell lung carcinoma, right? So small cell lung cancers are usually, if smokers will develop that, they do not respond to a lot to targeted therapy. Precision oncology doesn't have too much say in small cell lung cancer. But if it's non-small cell lung cancer, then lot many things have happened in the genome of this cancer type, and lot many things we know sequenced. So we know where all the mutations happen and what all therapies can be given, right? So it's a good news in a way, sort of. And the pathologist also advises that molecular tests should be done, and IHC, immunohistochemistry, should also be done. See, just to immunohistochemistry kind of segregates whether it's adenocarcinoma or squamous cell carcinoma. If just by H&E sectioning, if the pathologist is not able to tell, right? So then the patient, then it goes back to the medical oncologist and says that, okay, let's send the sample to the molecular lab. So how does the report of molecular lab look like? This is how it looks like, and this is an actual report actually. Um, I've just whitened the area which was showing the name of the patient. So it is it's non-small cell lung cancer, and if you see the mutations, so there are about 5 of them that we had captured. There was the gene, this is the gene name, so EGFR gene, ALK, so 3 mutations in ALK, FGFR3, PDGFRA. So these are just the names of the different genes, right? Of that, and this is the change in amino acid position. So this is where the mutation has happened, or I would say mutation has happened here, right? So if you're a molecular pathologist, you'll start, learning this, or molecular scientist, I would say, and you know where exactly the change has happened. And based on the change where it happened in EGFR gene or which gene, and based on what is the clinical history of this patient, you can start telling things. So I'll give you an example of this particular patient. So for example, here the change was in EGFR gene in L858R. This is the change, right? So amino acid L at the 858R position of the EGFR protein got changed to R. So what does that mean, right? So if you see so this is, now we are going into the molecular biology of cancers. So if you see at the cell level, so let's say this is a cell. So this is the plasma membrane, this outer covering of the cell. Then you have cytoplasm, and this is the nucleus. So EGFR protein is actually a transmembrane protein, which is lodged in the plasma membrane, right? And when the ligands of EGFR, let's say epidermal growth factor, EGF, it binds to EGFR. So, EGFR kind of changes the conformation, right? It changes the shape. And that change in shape triggers a phosphorylation reaction, right? So basically, if a biochemist would know better. So basically, a phosphate group from ATP is shifted to EGFR, one of the domains of EGFR, and then a relay race kind of starts. So the phosphorylation is transferred onto GRAB2, and then the phosphorylation is shifted to SOS, then RAS, RAF, MEK, ERK, and then the phosphorylated ERK molecule from cytoplasm, it will travel inside the nucleus, and it will eventually cause proliferation, right? So proliferation of cells happen, and this is a regular process, right? This is how cells multiply in our in our body, one of the ways, right? But the problem here in this patient is the EGFR changes its shape and triggers the entire cascade when EGF or the ligand binds to the EGFR, right? Only then this entire thing happens and the cell proliferates. In his case, EGFR protein, because of this mutation, is already locked in that changed conformation, right? So it's already locked in a conformation where it is active. So it is continuously triggering this signaling pathway, right, which is causing the proliferation continuously. And that is the cause of cancer. And that is why this cell has gone berserk. It is continuously multiplying. That is what is causing cancer. So what we can do about it? So then the drug companies, they have found, and we can go into details, but that there are molecules when you inject in patients or when you ask them to. So one good part of targeted therapies is that you can pop in as pills, just like paracetamol, you can just take that as pills. So you take it as pill and eventually it will, go from small intestine, absorbed, and finally in the peripheral circulation system goes to these lung cells. Inside the lung cells, they will go bind to EGFR protein, right? And they will lock the phosphorylation stage, right? So it cannot trigger the downstream signaling pathway system, right? So this won't happen. So you take this tablet and it will block EGFR, and so the downstream signaling pathway system blocks, and so the cell is not proliferating. So basically you are stopping the cells from multiplying, and it is going to block only those EGFR molecules which has this mutation. It will not block a normal EGFR molecule, right? So that's why it is targeted therapy, because it is targeting the cancer cells specifically, right? So if you go to FDA's site, it will tell you that, okay, this is the label, FDA label for this drug, which is Erisa, and actually Erisa is not very expensive. In India, patients are regularly getting this therapy when they have, stage 4 lung cancer. It is approved for EGFR exon 19 deletion mutation or L858R substitution mutation, right? So you can give Erisa. The dosage is 250 milligram. It is taken orally. uh, this is the, so if you see the dosage, it is, once daily, right? With or without food. This is the cost of Erisa. So 30 tablets will cost about 25,000, right? In Indian market. And if you go, it is one of the cheaper targeted therapies. So the one problem with targeted therapies are that they are not very cheap, right? But this is one of the cheaper ones, and this is the proof of the pudding. So for the same patient, this was the CT image of this big tumor, and just 3 months, of gefitinib, which is Eresa, the tumor really regresses, and this is another angle, and again it regresses. So this data is of Dr. Nirmal Rawat. This patient came to Dr. Nirmal Rawat, who practices in Mumbai and Thane. He's a very good friend, and we keep discussing a lot over patients, and because we used to do all the genomic analysis for him. So he used to share a lot of images. So one of the simplest case was this one, where EGFR, which is a known mutation, happened, and the drug was given, and you can see that within 3 months, the clinical response was pretty effective, right? So what I'll do is I'll stop here, and we can have a lot of questions, and I can also talk about what we do more in IIT. About, IIT is not uh, all about engineering actually. So it is a lot about medical sciences also, and I would also briefly touch upon this, that we have a very thriving medical technologies program where which runs between IIT Jodhpur and AIIMS Jodhpur, and half the students come after doing MBBS or MD, and half the students come after doing their engineering, either bachelor's or master's. So we run this program to build, medical technologies, which could be an assay, a very innovative assay, which could be a device, which could be sort of like computer vision model, or a lot of these things. And the reason why AIMS Jodhpur and IIT Jodhpur have come together is that these students can go to AIMS Jodhpur and they do something called as immersion program. So for an entire semester, 3 days, they just sit in AIMS, different OTs and clinics, cardiology to everything. They talk to patients and doctors to find real problems in clinic, and then they come back to the IIT faculties to find an engineering solution to these problems. So then one of the person becomes the guide from AIMS and one guide is from IIT. So then the student is also given an incubation center we have. So once the prototype is developed, we encourage the student to incubate their company and the faculty to incubate their company in our incubation center, actually take out the device to take it to the clinic. So in the real bench to bedside model, it also confirms according to the vision of our Honorable Prime Minister for Atmanirbhar Bharat, and AIMS and IITs are committed to that vision. So we got the third batch of students. Almost more than 100 faculty from both sides are involved in it, and our incubation center. So management faculties are also involved in it. Uh, Entrepreneurs are also involved in it. It's a very vibrant program. So I would like everybody at Baroda also to spread this word that we are running this interesting program. And if there is any student who is, whether medicine or engineering, who is very passionate about building in the space of medical technology, they can connect directly to me also. That's not a problem. I can give a lot of information. So any questions anybody has, I would be more than happy.
Thank you, Dr. Siddharth. Wonderful presentation. Can you stop the screen sharing? Because, yeah, that's good.
Okay.
Wonderful talk and very interesting things to know. I know a couple of persons in my family who wanted to do medicine but didn't get in those era, and now they are in engineering. So it's good to see the combination of engineering and medicine coming together for benefit of the patient. So it's open now. Jagdish Chinappa, you can come up with the comments. Dr. Jagdish Bhai first. Dr. Jagdish.
My question is, every cancer has a different genome. Every cancer will have a different genome.
So actually, sir, so is everybody comfortable Gujarati also?
No, please continue in English. We have people from outside Baroda also, and outside Gujarat also.
Okay. So the genome of every cell of our body is the same, right? So if you take a cell from brain to heart to, and if you just sequence the entire genome, it will be exactly the same. But the expression of genes in different tissues are different, and that is what makes every tissue different, right? So EGFR gene is one of the major cell growth factor in case of lungs, right? It may not be true for, let's say, prostate cancer or prostate cells, right? So genomes, while the genomes are identical, right? It's like the classroom. So the classroom is identical for every student, right? The benches and the fans and the ceiling and walls and the blackboard, everything is same. But one student is different from the other student, right? So similarly, like every cell of our body has same genome, but the expression of the genome, right? The gene expressions are different from tissue to tissue. That makes them different. And that is why the cancer basically manipulates one of the existing pathways and just converts these normal genes into oncogenes, right? When the mutation happens, it becomes from normal into an oncogene. And that's how the cancer is triggered.
Yeah, Dr. Jagdish Chinappa.
Good morning, sir. Thank you for a wonderful lecture and eye-opening, work that you're doing at Jodhpur. I have 4 questions, and I'm sorry if I'm going to ask them one by one. The first question I have is many patients who are put on this targeted treatment tend to develop what is called as they don't respond to the treatment after some time, after maybe a year or so. So what do you do then?
So basically, it's like in Hindi, there is a saying, uh, "Tu dal mein paat," right? So it's a continuous battle. Cancer is like a hydra, right? Hydra with 9 heads. So you keep chopping the heads and it will keep erupting new heads, right? So basically, cancer evolves. So if you read Siddharth Mukherjee's, he was an oncologist and wrote a very nice book, The Emperor of Melodies. So in a very philosophical sense, he said it is the
Yeah.
evolution that has been hijacked by cancer. So we evolve, right? So even cancer cells are, so in a way, cancer cells are more evolved. They dominate over existing cells, and so it is a normal process of evolution. And that's why when you treat the cancer with one therapy, it will find another way. It will, in the process of evolution, some of the cells that can escape this drug with because of a new mechanism, they will then start multiplying and they will come back again as a new cancer. So but don't mistake this as the relapse cancer is exactly same as what you encountered 2 years back. It is not the same. It is a new cancer. It has the imprint of the old one, but it is a new cancer. The reason it is not responding to your therapy now is because it has rejigged its molecular pathways. So that is why you need to take the relapsed tumor site and take out a biopsy and do a molecular analysis again to understand what changed. And with the changed knowledge, can you do a new therapy, right? So that is how it will go.
Thank you, sir. Thank you so much. The second question I had for you was many of these investigations are being sent abroad, and they take a lot of time to get back the report. So, are these facilities available at Jodhpur for doing the genomic analysis?
So you don't have to come all the way to Jodhpur for this. So I'll tell you some of the. So one, the nearest company is Newburgh Center for Genomic Medicine. It is housed in the old Supratech laboratory, right? It is one of the. So I had set up actually the solid tumor division. So I set up that vertical. Currently, Dr. Kunjal Patel is heading that department, and he's a very nice fellow. And so you can send there. They have the state-of-the-art machines like Illumina platforms and Thermo Fisher platforms, and you can just connect with Dr. Kunjal. And the other companies are in Bangalore. You have MedGenome, you have Strand Genomics in Bangalore. Also, SRL also works, but the nearest that you have is Newark Center of Genomic Medicine at Ahmedabad. There is also uh, uh, a company in Surat. Uh, I forgot the name, right? Dr. Salil Waniawala's. So that company is also, so, these are the places which are very close by to you. So you can get this test done all here. You don't need to send it abroad.
Dr. Jagdish, by the way, is from Bangalore. He's a pediatrician. One who is asking questions is Dr. Jagdish.
So Dr. Jagdish, you can access MedGenome and SRL.
Any other question?
No, actually, the reason is that we had. I have a close friend who has. I have a close friend who has got malignancy, lung malignancy, and we had to send his samples abroad for getting the genomic analysis. So that's why I asked this question. The third question I had was a little bit tricky, is about the ethics part of it. So in your opening statement, you said that if you have the doctor's registration number and the other number, you could access the genomic signature of this particular patient. Well, I suppose that the patient's consent also should be looked at, and especially if it is a child, it's probably more important to get a parental consent before looking at all this.
Absolutely. Actually, that is more of a futuristic scenario I was trying to paint. Yeah.
Okay. And the last point was, sir, please pass on your email ID and your contact, and also an invite to come to Jodhpur to see your center.
Yeah. Dr. Siddharth, you are muted. Please unmute.
Typing it right now, sir, so it'll be there. So
We missed your
that is my
answer.
email, and that's the phone number. So, even for your friend's case, I'll be happy. So, I also donned the hat of a molecular pathologist for quite some time, and one of the startups was also on molecular oncology. So, I really understand this domain very well. So, you can share the reports, and I could at least help. Knowing whether this sample should really go to US or whether it can be, resolved within India. So in US, there is a company called Foundation One, which is which is a pioneer, right? So one can send to Foundation One if the case is very tricky, right? But usually in lung carcinoma, very well worked out carcinoma, and most of the Indian companies are covering all those genes. And to be very honest, all the kits are coming from US only, right? So it's except for if you're sending to Foundation One, I don't see, the US company would use the same kit that an Indian company is using.
There is a
Right, sir.
question from Dr. Shulin. Any triggering factors found which are triggering normal genome to oncogene? I think smoking and all that will answer that question. He's interested in knowing the triggering factor
Yeah, I
for
can see the question
transformation.
in the chat box.
Yeah.
Yeah. So I think, these normal genome to oncogene, yes, like, cigarette smoke, pollutants, they are known to be triggering factors. So cigarette smoke will keep going inside the lung, and they will percolate through the cell membranes and go inside the carcinogens can then pass inside the nucleus. They'll start interacting with the DNA. That's where the change happens, right? So same thing you can talk about other pollutants. There are a lot of carcinogens which we do not know about, right? No study, no research has been very methodically done. So we do not know many times, things coming through our food. Many times, people say that, I was such a healthy lifestyle, ended up with cancer. How did that happen, right? So we really don't know from where we are ingesting these carcinogens, because regulatory bodies are also not very strict about, our food quality standards and a lot of other things, right? So some of them we know the triggers, and a lot of them we do not know. Yoga and pranayam, yes, I am not an authority. I haven't read a lot of literature about it, but I feel that there is a lot of wisdom in our ancient system, and it may have an effect. Probably somebody has to do research on actually proving it in the modern scientific method, because nowadays unless you prove it in by modern scientific method, nobody will accept the proof. So if somebody does that, then yes.
Hey,
Siddharth,
Yes, sir.
the world's largest private cancer center, Memorial Sloan Kettering Center, has a website, MSKCC, and there is a huge section on herbs where all the scientific data of the branch of genetics where the lifestyle and diet and external factors change the genes is called epigenetics. Now, that is a big topic. We have to understand it in a next lecture. Correct, Siddharth? And all the oncological genetics tests are done in Vadodara. There is a person like you in Vadodara, Mr. Chaudhary, who has started his own molecular biology lab with all the facilities. And all the oncologists, medical oncologists, especially in Vadodara, they have connections with the best genetic laboratory. We have even appointed a geneticist for each oncology centers. So Vadodara or Ahmedabad or Gujarat or India is not behind any center. Next point is,
Absolutely. Yeah.
for all the clinicians to understand this, your very complicated topic of carcinogenesis and genetics, there is a simple paper called Hallmarks of Cancer by Gallagher. Correct? But that gives you the 12 heads of Hydra. So I suggest to the audience that hallmark of cancer should be searched, and they should read it, that, and it will be clear how modern genetics. Third point is that all the targeted therapy are done before, are done only after a molecular geneticist like Siddharth will say that this is a ALK positive or EGFR positive. So molecular biology is guiding. My question is about prevention. Molecular biology, has it translated into a routine test where prevention occurs, and that is occurring in all the genetically susceptible families like Lynch syndrome, and there are 15-20 syndromes where Siddharth, like geneticists, have identified that if this is there, then likelihood of cancer is more.
So I'll just answer the last question. So basically, treating beforehand, or without giving these therapies, targeted therapies, gene therapy is possible. So CRISPR-Cas9 systems, but they are still in the research papers, and very very minute part of it has come in the clinical trials, right? But the scientists are really in that process of trying to like, for example, inherited disorders, genetic disorders. So right now we can just counsel, we can identify the disorder, and we can say change the lifestyles, but we really cannot treat the patient. So once a gene therapy becomes a reality, then there is a hope that such patients can be actually be treated. But it's a long slog. I'm not, I mean, this is just and I'm not even a very I don't know a lot of literature on about gene therapies,
But
but it is there. That much
But
I
the
can just say.
simplest first step has been taken in leukemias. CAR T therapy, where the blood of the susceptible person is taken, genetically altered, and all the leukocytes are put back into the cells. It is an expensive therapy. Few centers in India have tried it, and it is a landmark therapy that is helping. Genetics is helping directly.
Thank you, Dr. Sant. I have to inform the audience that Dr. Siddharth has to leave by 8:45. He has some flight to catch. So we don't have many minutes now left with us. I would request Dr. Radhika to make any comments, because let me acknowledge that it was Radhika's efforts which got us Siddharth with us today morning. So if Radhika is around, Radhika, you can make some comments and then we can end the meeting.
Yeah, I can see Radhika.
Yes.
Hi, Radhika.
Please unmute Radhika.
Yes, so mine is going to be a non-scientific comment. I just wanted to let you know the background of Siddharth and I. So we both are students of MSU, and Siddharth taught me molecular biology. Very honestly.
Oh, is it? No, that's not correct. That's really not correct. I think
the old days when we used to sit with Heniatis, the thick molecular biology Bible, and he would read it page by page, find us protocols, teach us everything. He was senior in the lab, in Professor Naresh Kumar's lab, and I was doing my thesis work in that lab, my master's thesis, before I went off to US and before he went off to US also. But so that is just the background. I wanted to tell you that we grew up here in Baroda and have gone in many directions, but we've stayed in touch in many scientific ways and other ways.
Yeah.
So good
Thank
to
you,
have
Radhika.
you.
Thank you, Dr. Radhika, for getting us such a nice speaker, and we look forward to some more talks in future, Dr. Siddharth. This is a very evolved group, I would say. The Saturday Club group running for more than 50 years under leadership of Dr. Satisa, who is there in the frame, you can see. And this is a very evolved audience in terms of very eager, very hungry for knowledge. So these kind of talks make us very happy to listen to. Little bit of the routine things that we usually listen. This was wonderful, and thank you for being with us. We'll definitely invite you for some more talk in future. So thank you so much. Thank you.
Thank you so much, Pandya sir, and thank you so much, Radhika. So connecting me and
Satish bhai, I have one question just before we leave.
Sir, yes, I mean, sir.
Yeah, methylation profiling, what is the role?
Siddharth, can you hear?
Please repeat, sir. I mean, sir.
No.
There is lots of knowledge about it. Methylation profiling.
Methylation, DNA methylation.
So that actually, what you talked about epigenetics, methylation profiling is also done right here. Especially, if you see colon cancers, you will find methylation profiling is done on a regular basis in again stage 4 colon cancers. So before giving certain kind of therapies, methylation profiling is done. So it is, that again brings to, without making a change in the DNA sequence, you can still make a change by just, hypermethylation. So, I think a lot of this is there as literature. A lot of this is also there in the clinical aspect. But you are absolutely correct, sir, that this is happening as part of genomic medicine. Actually, you have a resident expert in Dr. Radhika, so she can just take you through all these. For her, this would be child's play, whatever I blabbered here.
Definitely, we'll take that clue from you. We already had her as giving us one talk on CRISPR technology, if I remember correctly, right, Radhika?
Correct.
So
Yes.
we end the meeting here because Dr. Siddharth had requested me to end by 8:45. And I think we should end the meeting here so that he can carry on with his work. So thank you all.
Thank you so much, sir.
We meet
Thank you so much.
again next Saturday. Thank you all.